Hello and welcome to the Xapiens Podcast. I'm your host, Loyd Waites. During the COVID pandemic, pharmaceutical testing was suddenly brought into the limelight. People finally began to realize how expensive and time-consuming pharmaceutical testing really was. Now imagine if you were able to replace an animal testing system that costs billions of dollars and most of the time fails with an in vitro human cell system that could be used to develop drugs faster, cheaper, and more reliably. This is exactly the work that Donald Ingber and his group does, and later they tested during the pandemic. I talked to Professor Donald Ingber about this during my interview today, and I realized that this only scratches the surface of all of the amazing work and the process that's done at the Wyss Institute that ranges all the way from genetics to robotics. There's a really great thing that we did today with covering lots of different interesting information. I hope you enjoy it. Thank you. So thanks for coming out to talk with us today.
My pleasure.
Yeah. I've seen a lot about your research. I've been following Emulate pretty closely for a couple of years now. Would you mind giving just a brief research overview of a lot of the stuff that you've worked on and that you're working on now?
Whew. I've been working over 40 years. So my work has always had a mechanical, physical bent to it, even though I'm a cell biologist and MD by training. As a graduate student, I basically proposed that mechanical forces are as important as chemicals and genes for development, diseases like cancer and so forth. And that has sort of been a core part of everything I do. The past 13 years, I'm the founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard. And that is kind of an amazing place in that the concept is that engineering's transformed medicine and non-medical areas over the past 50 years by taking engineering principles, trying to solve problems in medicine, things like hip implants, drug delivery, pacemakers, stents, etc., as well as environment like manufacturing. But we really believe that we've uncovered enough about how nature builds, controls, and manufactures from the nanoscale up that we're literally at a point where we could leverage biological principles to develop new engineering innovations. That's what we call biologically inspired engineering. So, I probably have the largest group, largest funding at the Institute. But the Institute, involves people like George Church, Jim Collins, Jennifer Lewis in 3D printing, David Walt, who founded Illumina, Dave Waites, Samir Michigotri in drug delivery. It's really quite an amazing place. So big synthetic biology efforts, sustainability efforts. My own work, major part of it has been what's called human organs on chips. These are microfluidic devices that have lined by living cells that are culture devices, but we literally can form tissue-tissue interfaces, which means we're developing models of organs, not just cells or tissues. We have fluid flow. If it's a lung, we could have air-liquid interface. We can have breathing motions, peristaltic motions, and with that, we've been able to recapitulate both healthy and disease states. We've linked them together to create what's a human body on chips. We could predict drug pharmacokinetics. So with that part of my effort, we are really focused now on leveraging that for repurposing existing drugs, particularly around COVID, very quickly that could be used for new applications. So you could skip the long development process and quickly try to initiate clinical trials around a specific use. They're already FDA approved. And then also drug discovery, because we basically have the ability to do human experimentation in vitro. And we combine that with AI, machine learning types of approaches to take the huge amount of data from transcriptomic databases and basically prioritize drugs that we think would have a high likelihood of reversing a disease state to normal based on clinical transcriptomic data. Then we could test these in the chips. And we also have been have interesting recent work where we do that. We find an existing drug that seems to work. For example, in COVID, we found one that we targeted a region of the spike protein on COVID virus, or SARS-CoV-2, that's highly conserved. It's not where all the antibodies target. It's not where your immune system usually targets. It's sort of hidden, but being biophysically, focused, we looked for what needs to be a pivot point to open the molecule and go through large-scale molecular changes for membrane fusion. And we used our computer AI machine learning approaches to identify existing drugs. We found an existing drug, oral drug, well-tolerated, pretty good efficacy, but not what would be optimal. But then we used molecular dynamics simulation, which is how we found the drug, to basically design new, and chemists and medicinal chemists, and we design new versions of it that now inhibit at really low, in the nanomolar range, every variant, including Omicron that we've tested, as well as other forms of coronavirus. So those are examples. I can give you many more. But the other thing we're doing that came out of Oregon CHIPS was we were funded early on with a major grant from DARPA, something like a $36 million grant, to develop 10 different organs and link them together to create this human body on chips. But we were also funded to develop inline sensors to be able to read out functions. And that led to a whole diagnostics effort where we've now developed electrochemical sensors, sort of like a glucometer for people with diabetes. It's a little blood stick, and it's a quantitative readout, digital readout, disposable chips, cheap, mass producible. But we've developed ways to multiplex it, so you could do like 10 or more analytes at the same time in minutes, electrochemically. And so that has now been licensed to a startup. It's been licensed to a company that's starting COVID trials. You could do multiple analytes at once, like we have a paper now where we're doing CRISPR-based detection of viral RNA at the same time as antigens, at the same time as host antibodies. So you could detect where they are in their response to infection and immunization. So I now lead a platform that's called bio-inspired therapeutics and diagnostics. And there's many other things that have come out of that, but those are examples of what's going on right now.
Okay, that's super interesting. I have so many questions already just based on what you were talking about. So I think the first one would be, there's a lot of different organ-on-chip groups out there that are spread all over the world, many in Europe as well as some here in Cambridge. What do you think makes the emulate chips in your organ-on-chip systems stand out more?
Yeah, and we use commercial emulate systems now. So I think one thing is that they're commercialized, so you can get the same chip every time. So not in the early days when we started this, each grad student or postdoc made chips that were a little bit different and a little bit less efficient. We also had pumps and tubing, and now this is a plug and play. So that helps robustness and reproducibility. What makes our systems different than other ones is that we use pretty much the same device for all different organs. So it's a common platform. The biggest difference is that we incorporate the mechanical microenvironment. So we could actually have breathing motions for lung and peristalsis in addition to flow. Now, you might not think that that's important, but it actually is all important in terms of both replicating normal physiology, but also disease states. And then most amazingly, a recent success paper that's just about to come out, we found with viral infection, with influenza, that, and which we used as a model for respiratory pandemic viruses like COVID, we found that the breathing motions on chip regulated the efficiency of viral infection. And so that you basically, healthy breathing suppressed infection. It worked it through the innate immune system. We did mechanistic studies, worked out the pathways. We found a target called RAGE. We tested a drug called Aziliragon that has been tested in 2,000 patients, oral drug, very safe. It was being tested for inflammation in Alzheimer's disease. Didn't quite get the robust response they wanted in Alzheimer's disease, but was safe. In our chips, it completely prevented the cytokine storm. And now that's been shown in a hamster model by another group. And so those data, based really on breathing, it would never have been found in any other chip. That was just submitted to the FDA for an investigational new drug to start a COVID trial. So that's an example where breathing, where this does matter. Another novel feature is that because we have two channels and we have this tissue, we have a vascular channel with endothelium, and we have a parallel channel that's interstitial, if you like, we can grow complex human microbiome, which is like one of the big paradigm shifts in medicine in the last 20 years, which almost is always analyzed in terms of genomic, metagenomic correlations, if you like, guilt by association. We can now take a stool sample, take all the microbes, put them in an intestine chip with peristalsis and flow, and keep the chip alive and happy for 5 days and study host microbiome interaction. We're doing this with vagina, we've done this with lung. So those are other really, I think, novel features. People, I had the first big breakthrough paper where it was really organ on chip term, and they use it for everything now. And I mean, they literally use it for anything that is more complex than two-dimensional cells in a dish. But I really use it for microfluidic systems, and there are other style systems, but I think ours certainly has been shown the highest level of clinical mimicry again and again in many different organ systems. So those would be the biggest advantages or differences.
Yeah. Another thing that I guess when I typically describe these systems to other people, I always say, well, this is this new form of testing for pharmaceuticals that is faster and cheaper and more reproducible than a lot of the kind of current paradigm, but it isn't quite there with FDA approval yet. But I also know this is changing.
Yeah, it is changing. So the first thing is that the most important thing is that it's more likely to predict human responses. Because the huge cost is not buying animals and doing the study. The huge cost is having a drug that goes through that and all the clinical stages and tens of millions of dollars, and then it fails, which is 70 to 95% of the time. So that is really the advantage of these chips. The FDA doesn't approve things like this generally. They approve a drug. But what they do is have ways to recognize that they'll accept certain test systems in lieu of animal models. Like an example years ago was they developed a biochemical test for kidney toxicity, and they were able to apply to the FDA and get letters saying, we will now accept this test instead of this animal assay. Now, based on the huge, burgeoning field of organs on chips and other what are called microphysiological systems, the FDA has started a new application route called iSTAND to enable that sort of thing. So emulate, we have a paper that's in review that is a preprint on where they did 780 human liver chips. And again, often when people say they have a liver chip, they have, even if it's microfluidic, they have like liver cells in a chip. Maybe they have liver cells and endothelial cells. These chips have primary human liver hepatocytes, primary human liver endothelium, primary liver Kupffer cells, and stellate cells, which are other sort of cells involved in inflammation and responses. So 4 different cell types, 780 of them. They tested 27 different drugs that had been identified by a consortium of pharmaceutical biotech companies as benchmarks for being able to be better than animal models, because they have all the data of what animals said would be safe and not safe, and then what really happened in humans. And basically, the net sum is that the study showed 87% sensitivity and 100% specificity, which is far better than 7 or 8 times better than animal prediction. So that they're applying to iSTAND. So is it approved yet? No. Are they going to ask for more? Probably. But I think we're getting closer. Is the answer.
Yeah, that's really encouraging to
hear. So I will say that. Plus, we just first time ever submitted an IND with data from organ chips in it. We have drugs that are in clinical trials, partly based on data we obtained in organ chips around COVID in Africa that we repurposed to drug. We're getting closer.
Yeah, that's great to hear. I've also heard that because it's replacing these animal models, that you get a lot of support from animal rights groups in terms.
Yeah.
And so, how do you feel about that?
Oh, it's wonderful. I mean, I'm an animal lover, so when I first had the 2010 paper, I had an old cell phone before iPhones, and it was set so that it would beep when I got an email, and it was it took me a long time to figure out how to turn it off because people were so excited about it, and always are. I travel around the world. It's one of these things where it's visual, it the images on the chips are gorgeous, and then the fact that we're potentially saving animal lives, it's a real crowd pleaser. It's really obviously gratifying. And this is a true story. I did my MD PhD at Yale. I had done a lot of animal work, and I'm a real animal lover, and I hated it. So when I came to Harvard to do my postdoc with Judah Folkman, and he did a lot of animal work, I didn't. I got involved in little bits, but I really worked on developing in vitro models. It was way before organ chips, and there were different models. Each one had novelty, but kind of in the back of my head, that was always a goal. So I didn't do it thinking I'm going to go replace animal ever, but I did think I don't want to be working sacrificing animals on a personal basis.
Yeah, that's great to hear. I'm personally a big animal lover myself too, so that's good to hear. But it's not just about that too. There's also an aspect of personalized medicine that can be used here as well.
Absolutely.
Right?
We had a paper recently where we did a bone marrow chip with cells from patients' marrow or blood, and we can get complete white and red blood cell formation for a month on chips. We can show similar, and this is very important, we could show similar sensitivity to drugs and radiation at the same dose exposure in humans. So when you hear about people testing drugs in a dish, they will bathe the cell in the drug for days. And so let's say you're looking at marrow toxicity. They'll grow marrow cells in a dish. They use a high concentration of drug. 5 days, they see toxicity, and they say, oh, we mimicked a human response because it causes. The fact is that in our body, the patients who show those toxicities have a particular dose range that's infused, IV for 2 days, in this case for a cancer drug called 5-FU at 4 micromolar for 2 days, and they see a toxicity. If you give it at 4 micromolar, bathe it, you don't see anything. But if you do it in the chip, perfuse it, like an infusion for 4 micromolar, you see exactly the same thing. We had a company, AstraZeneca, come to us with a drug that they found in human clinical trials, and it's really unusual for a pharma to do work with academics when they have a drug in clinical trials, but they had what's called a regimen-specific toxicity, meaning same dose, 2 different time frames gave different toxicity. So same dose over 2 days gave low, I think it was low white blood cells or red blood cells, I can't remember. Over 2 hours, it was both. And they couldn't really replicate that easily in animals, but we replicated that beautifully on chips, and we were able to precisely mimic the change in drug levels over time in these patients, because they've been given, give it 2 hours, it goes up, and then it comes down quickly. Over 48, it's up high, and then it comes down. Well, they had measured the levels in blood of the drug using mass spec, and this is called pharmacokinetics, PK. And so they had the PK graphs. When we flowed through the chip, we beautifully mimicked that graph, and we beautifully mimicked the toxicity. This is a level of clinical mimicry and fidelity of recapitulation you really can't get with other systems without microfluidics. That same study, to come back to your question about personalized medicine, we made chips with cells from children with a rare blood disorder called Shwachman-Diamond syndrome, SDS, and they usually have decreased blood formation, and we saw all that on chips, which is cool. But then we were actually able to uncover that we saw a defect in a white blood cell maturation that wasn't really known before. And we went back with a lot of patients, and it turned out that 50% have this defect, 50% don't. So we begin to get insight into etiology of the disease. But the idea that you can take a rare genetic disorder where there may not be enough people to develop a drug or to do a clinical trial because they're all around the world, but get their cells, make 50 chips, find the optimal drug combo, either existing or new for them, and then do a clinical trial. That opens up that sort of possibility. Or to do male versus female. We're funded by the FDA right now on radiation toxicity. We're looking at male versus female marrow. Children versus adults. It's very hard to do pediatric trials. You can get cells from kids and do them on chips and maybe shortcut things. So yeah, it has major implications for that.
I've also seen in one of the talks over at Emulate that there were 2 major paradigms that the group was kind of working to change. The first being drugs that
failed clinical trials due to being inaccurately represented in animal models, and also allowing for drugs that normally would never be able to go on the market because they didn't make it past these animal trials, but also evaluating new drugs in a faster way.
Yep.
Can you speak to those kind of?
Yeah, I mean, I think the liver toxicity is a great example where there are a lot of good drugs that are taken out of the line because you have a rat and a dog, and the rat looks good, but the dog looks bad, and that's enough to kill it, even though neither of them mimic human. Okay, right. So that's a great example of that side. I think there's so much time done in these preclinical models that don't correlate with human that if you can go right to human and you can get a feeling for safety and efficacy, you are shortcutting the process, and it's a huge potential saving. That liver chip study on toxicity with a economist, they emulated an analysis, and I think just from that one model, they estimated a savings of $3 billion for the pharma industry, not because of the cost of the animals, but because of the savings of not failing.
Yeah, and I think I heard one story about a test that was fine in animals, and then when it went on to clinical studies, actually caused the deaths of some of the.
Oh yeah, there was a drug that caused. It was a therapeutic monoclonal for lupus that had no problem in animals, and it caused pulmonary thrombosis, clots in the lungs in clinical trials. And I can't remember if there were deaths, but there was major morbidity, and it was pulled. That we could mimic on chips.
Right. Yeah. So what do you think is kind of just around the corner for this? I mean, you mentioned full human body on chip with multiple different organs wired together microfluidically. Do you have?
So it's interesting. We did that. We did 8 or 10 different organs. We linked together. I think it's more likely going to be just 2 or 3. Like we linked what's called a first pass model of intestine, liver, kidney, because that's really where if you have an oral drug, it's absorbed, it's metabolized by your liver, it's peed out by the kidney. We had a little mixing chamber so that you're not taking samples right after each chip, but something like your heart mixed it, and now you can take a peripheral blood sample. And we, with computational modeling combined, like measuring the drug levels in each chip, and then using computational modeling of what's called PKPD, pharmacokinetics, pharmacodynamics, that is used clinically. But clinically, they take into account the size of the liver, the flow rates, the flow volume. We scaled that down to these little chips, or scale our chips up to that computationally, and we could quantitatively predict the pharmacokinetic dynamics of these drugs using chips. So that's an interesting possibility. Again, shortcutting a lot of studies. I think the whole body on chips is going to be it's really hard to do many chips at once. You have to get each one of these chips cultured just the right time and link them together. But I do think like 1 or 2 chips where you're trying to say, is this drug metabolized by the liver and having toxicities, making sure it's not, that sort of thing will be done. I think the clearance functions, the PK modeling, we have developed an immune chip. This is about to come out. That is a lymph node chip, which basically you could literally vaccinate with a commercial flu vaccine and you get the entire vaccination response. So right now they use non-human primates for that as major ethical issues, major shortage. So that's really an exciting one. I think animal lovers would be very excited about, but also can a different way to affect the therapeutics development pipeline. It's not a drug per se. And you go to human right away. And there are many cases where even non-human primate results do not predict what happens in humans with vaccines. Many cases. So, I think it affects all facets. It's also important for toxin testing, environmental toxins. People have wanted to make, can you have a cow chip? Can you have an insect chip? In theory, you can. One group is working on a bat chip to look at the One Health challenge of these vectors being created in animals that are you don't conventionally culture in the lab, and you can't culture everyone, right? So that's really interesting, I think. Because if you could stop infections before they get to human, that's really the way to do it. So it opens up entire range of possibilities.
Yeah, I think this was another organ on chip system that I had seen that they were essentially accelerating the mutation process of viruses by running them through multiple different chips over and over and over again.
Yeah, that was my group too. Yeah, so we culture with influenza in an airway chip. We use a drug that at 90% efficiency, so you're missing some of the viruses. And then we do it for a few days, and then we put a little droplet of fluid through the airspace, and we pass it to another chip's airspace, just like a respiratory droplet, in the presence of the same drug at 90% efficiency. And we do this multiple times. We're not speeding up evolution. This is the way viruses evolve. We're modeling human-to-human transmission and emergence of variants. With one drug, which used to be a first-line drug many years ago, but isn't anymore because of resistance, called amantadine, 8 passages, we got resistant virus. But what was amazing is we did genome sequencing, and we found all of the known reported mutations that have been identified in humans who became resistant. But we identified, I think, 3 or 4 new ones no one's ever seen before. So that means you might be able to see what's coming down the pike, which is, we have to guess on what virus is happening next year, or look what happened with SARS-CoV-2. So you can develop therapeutics ahead of time. Now, we did that with the first-line drug. It took 25 passages, but we got resistant viruses, which is really scary. We even got, we also saw that this wasn't just mutation, this is also reassortment. If you did 2 different viruses at the same time, they would reassort to become resistant. So it mimics what you see in humans. So that opens up another type of investigation, which is incredible. In terms of, you asked about the different types of drug development. We did stuff with microbiome where we could use metabolomics to, like measure thousands of different chemicals at once with mass spec and identify specific chemicals produced by the bacteria in the gut that, in this case, modulated sensitivity to infection by enterohemorrhagic E. coli, so a bacterial infection. It's well known that mice are incredibly resistant to this bug, and humans are incredibly sensitive. And there was suspicion that microbiome contributed. And we could show that if you take mouse microbiome and human microbiome, it actually is responsible. We were hoping we would find what protects the mouse and have a therapeutic. What we found is what makes the human more susceptible. But you can, and we did that in a couple of months. Wow. So I think for the discovery, and we did in months this drug that is now going to clinical trials for cytokine storm. So months. Everything takes a year, but the actual finding of it takes months. Publishing it takes 1 to 2 years. That's the real world right now. Yeah. Thank God for preprints because, the preprint came out a year before this drug, going to the FDA for an IND approval. The drugs that are in Africa in clinical trials, it might have been a year and a half before. No, I guess it was that one was fast. It was 6 months before they started to go towards clinical trial, but it was way before the publications came out, which is a whole other challenge in science right now.
Yeah, I understand that being in particle physics, preprints are the world. Because, because
it's gonna happen. It's gonna take time, but it's got to happen because it's crazy.
Yeah. I just want to say, and kind of like take a step back and say how amazing that it is that using the same technology that can be used for development of drugs in a cheap and efficient way, you can also have a hardware prediction of virus mutation. I don't think I've ever heard of another group doing something like that. Computational modeling, sure. But
people have done some selections in vitro. I don't know if anyone's ever shown that this was due to host virus interaction. And, we have mucus there. And demonstrated the level of mimicry in terms of these mutations that we see. But it does open up a new path to study those sorts of phenomena without having to do it in, hundreds of thousands of people.
Yeah, so what do you think kind of the limit for these organ-on-chip systems is?
The challenges are they're not high throughput right now, and they're high content, so it's more like an animal model. And so there are people developing higher throughput systems that may not have the full fidelity of recapitulation of human, but are great for first. I see this being a stepwise process that you use these as sort of in place of the animal and almost as like an early clinical validation. So that's one challenge in the field. I think the patient-specific chips, or what I think is more realistic, is genetic subpopulation specific chips, or people with particular like comorbidities. In other words, it's well known that people who have chronic obstructive pulmonary disease are much more susceptible to influenza infection, right? We replicate that on chips. So if you're testing drugs, you might want to actually, and like I was on the phone, I was on the Zoom with BARDA yesterday, which, is for the protection against, all sorts of threats, right? And bio threats and radiation. And they even raised this, could you do that with COPD? And the answer, we've done it with COPD, because they have to stockpile drugs. But if you know that there are different subpopulations you might need that have different response, you may have to have different drugs. So things like that, where in a clinical trial, most of these companies spend a huge amount of money. They do a big trial. They almost always fail. Then they do statistical number crunching to find out if there's a different genetic subgroup or a subpopulation that responded better. And they'll go back to do a narrow trial, and they get approved for a narrow application. With these chips, you can imagine like setting up different subgroups of population, whether it's COPD versus healthy, or, in my talks I always say like, Hispanic women with asthma who are ultra sensitive to cigarette smoke, but you only take 50 or 100 of these people, and then you do their chips and you find the optimal drugs, and then you use those same patients for your clinical trial. It could, shorten the process, decrease the cost, increase the likelihood of success. It could revolutionize pharma biotech. It really, really could. Is it going to happen? I think it will, but will it take a long time? Because nobody wants to change the way they do things. This is why you have startups. I mean, you have to have disruption.
So when you say low throughput, what do you mean? Do you mean few numbers of drugs being able to be tested at a time?
Yeah, I mean, right now, like the Emulate system, you can do 12 chips. Each one has one channel, so each one is like a sample of one. And so the pharma has incredible capability to test chemicals 100,000 at once against binding something, but the relevance of that is nothing compared to doing it in the context of inside a cell. And the relevance of that is nothing inside a tissue, and the relevance of that is nothing inside a tissue with other tissues around it and the right physical environment, which is what the organ chips. So you're going down in throughput, but you're going up in value. But people are developing higher throughput screens that are microfluidic now, so that's another opportunity. So far, they don't really have the mechanical side of it, but I think that's a real high value area to explore. People often raise the challenge of the materials used in organ chips. We use a polydimethylsiloxane, a silicon rubber, and it's great because it's clear, so you have like a window on molecular scale activities inside cells, inside a tissue organ contact. It's great because it's flexible, so you can do breathing. It's great because it lets gases pass. You get oxygenation. But it absorbs some small organic molecules like fat-soluble molecules, and so some drugs are like that. So that has been raised as a major, major issue. We've not found it to be an issue as long as you measure drug levels in these chips. But coming up with new materials for chips that do everything that I said, but also don't absorb any drugs. We've published some papers, other people are working on that. And of course the challenge to commercialize all this, right? That's the big thing. This is not going to be academics who change the world with it. It has to be companies, because you have to have the same results in labs everywhere, which means you have to mass produce these chips so that the same high specs are met, specifications. You have to have control systems that are automated, not everybody different. I think those more and more needs to be automated, not only in the running of them, but the plating of the cells, the analysis, more inline sensors. These are the kinds of challenges that the field is facing now. And then there's the commercial challenge of convincing people to change, which is the ultimate challenge.
Yeah. So I know you're saying that there's a throughput problem, but this seems like a very parallelizable system, right?
Yeah, you could have many at once. Right. Yeah, I mean, the Emulate system is 12 chips in a small unit, and you can fit like 3 to 4 in an incubator. So let's say 48 in an incubator. It's just, people are now, because you can do these microfluidics, they could try to make many in a small area, which is good, but then you have even less cells. So now you have such small sample that it's hard to have a sensitivity to analyze what's going on. So you're playing off these two different parameters.
Right. But if you want to just throw money at the system, you could have multiple incubators.
Well, that's how Emulate did 780 chips. They have a lot of systems, obviously. And it may be that they have to do that as a service for certain types of activities in the beginning.
So you mentioned the really high failure rate in a lot of these pharmacological models. About what percent is that?
In some areas, it's like 95.
Wow.
Others, it's like 70. If you look at the literature, and this is, FDA, NIH, pharma, they usually say 70 to 95%, depending on the area.
Wow.
And the stage in the pipeline.
And so do you think Emulate would more be working towards trying to fail often to succeed sooner, or to have a better model, or both?
I think what pharma needs is to know very quickly if, they don't want to lose things that are good, and they don't want to spend too much time on things that are bad, right? So go, no-go types of decisions would be good. But you have to realize that most people think these companies just screen and develop a drug, but they'll come up with a priority list of drugs. They'll have a lead. They'll move it along, spending huge amounts of money. They'll find a toxicity in an animal model, let's say. And then it's like, we spend all this money on this drug. Is that real? Why is that? Are one of our other drugs in the lineup better? And these chips are great for not only analyzing mechanism of biological mechanisms to study new drug development, but toxicity mechanisms. So you can use these now to go back and say, well, this is how that works, and these other drugs are better. And like we found that there's drugs that were found to cause liver toxicity. Turns out one of them didn't target the liver cell; it targeted the blood vessel cell in the liver, and that wasn't even known before. So it can really give you new insights. It also can be used to screen drug delivery systems, like we've done adenoviral vectors for gene therapies in chips. I think mRNA therapies people are studying. We're working on CRISPR-based therapeutics with DARPA funding right now on our lung chips for COVID. So that's a whole other side of it, is you can explore drug delivery efficiencies in a much more relevant environment where you have mucus, you have flow, you have air-liquid interfaces, not just cells in a dish.
Yeah. So what about like longer-term diseases? Are you able to keep the cells in the chips alive long enough to do like chronic studies, or is it mostly just acute studies?
So it's, we generally don't go beyond a month, just because it's, some of it is robustness, some of it is just, we have to get experimental results. But there's a funding mechanism out right now trying to support long-term survival of chips. I think you can get, we've got some of them go to like 2 months that we've pursued, but what's amazing is if you take cells from patients with a chronic disease, there are what are probably epigenetic changes, is that they retain the phenotype. So we have cells, just the epithelial cells from chronic obstructive pulmonary disease, they have features of chronic obstructive pulmonary disease, including certain immune pathways, propensity, ultrasensitive to viral infection, ultrasensitive to cigarette smoke. You can model exacerbations. If you take cells from a cystic fibrosis patient, the same thing. We model all of the baseline high levels of inflammation, the thick mucus, the increased bacterial growth. We found this again and again and again. So you can study chronic disease without having to do, long-term studies. Now, if you want to talk about long-term exposures, that's harder to do right now. Will people figure out how to do it? Probably, but Emulate and other companies have put instruments up in the space station for NASA, right, where they're totally automated and going, and so you can keep them longer. We tend to want to use primary cells because we think they're much more reminiscent of in vivo, which they are. But there are cell lines that probably can grow much, much longer in these chips. And often, if you have a particular phenotype, they often, if you confirm they replicate, that facet of the phenotype can be used to probably get longer term survival as well.
So you've talked about using computational approaches, and I'm assuming some machine learning approaches as well. Is that a fair assumption?
Yeah, we use sort of Bayesian network analysis as well, combined with machine learning. We've integrated machine learning with our molecular dynamics simulations. So yeah, I mean, various approaches.
Do you think things like that could help extrapolate to these processes that you're struggling with, like long-time exposures? Or
you mean whether you can circumvent some of the modeling computationally?
Right.
I think the more and more data that's out there now, which is expanding enormously, we can use that data to come up with things to test, and the test doesn't have to be. In other words, you use the long-term exposure in real patients. We just applied for a grant where DARPA wants to know, can you predict whether you're going to get a long-term strong or weak vaccination response by doing measurements in the first few days after vaccination. And we think you could do that. And we have these chips where we get vaccination response for a month. And the point is, usually that initial response sets up the long-term response. So there are ways to do these. I think probably, not probably. I mean, we've done stuff with diseases that are chronic using transcriptomics from patients and then come up with machine learning and come up with predictions of drugs. We had a really cool project where DARPA put out an announcement to develop drugs that induce a state of suspended animation, like true Star Trek stuff.
Yeah.
And there's no pathway to start with, so it's not like the way pharma would approach it, finding a molecular target. And they wanted to we basically combined organ chips with we have a very cool system I call organism on a chip, which is we have microfluidic devices that are in the shape of a peace sign. Okay. And we have tadpoles, xenopus frog tadpoles swimming in them. So it's like an infinity tadpool, the team calls it. So they just swim endlessly. But we have electrodes and colored lights. And it turns out tadpoles have a much more sophisticated central nervous system and immune system than zebrafish or C. elegans. But you could do high throughput. We can do 1,100 of these in these tadpoles. Each one's sort of on a USB stick. Each one is monitored continuously for motion. Oh, wow. And we can do operant conditioning. So you could give a shock when they go left, so they go right, and you train them, and you can measure learning and memory, and Mike Levin says frog IQ essentially, and you could also do CRISPR on these, so you could quickly do genetic models of diseases in like weeks, 2 weeks. You can do a CRISPR model. Oh, wow. Anyway, a startup just came out on this because with that, I'm digressing, but I will say, let me go back. So with the frog model, with the organ chips, with machine learning, we were able to, studying transcriptomics from organisms that go into hibernation or torpor, Alaskan ground squirrels, things like that. We able to come up with predictions to test some drugs, and in a period of like under a year, we had drugs that slow metabolism, which is what you basically want to do at the cell level, tissue level, organ level. And they now have slowed metabolism in a whole pig leg transplant at the military, U.S. Air Force in San Antonio, in San Antonio, where they have them in a perfusion chamber, and they can measure the ins and outs and oxygen. These drugs just slows metabolism like you're chilling the leg, which is what they wanted, so that you could have a soldier injured and maybe give them a chemical and get them off the field, or transplant a heart, or store your CAR T cells. Anyway, so it's amazing what you can do by combining with this AI approaches with these cellular systems. And what we did with the CRISPR, though, is we've made models of a genetic disorder that causes autism in children called Rett syndrome. And there, when you CRISPR out the gene that causes it in children, and in children they get like repetitive motions and seizures and also GI symptoms. It's amazing that the little tadpoles that normally swim in a regular way start spinning around like this, or they just lie there in a seizure. They also have abnormal ciliary motion, which is what's responsible for GI tract. And using the computational approaches with these high throughput screens with the tadpoles, we came up with drugs that work better than the best drug out there in phase 3 clinical trials in the same model, mouse model that was used to bring it there. That's now a startup called Unravel. So we're combining, I totally believe it, that combining the AI approaches with multi-omics, in an iterative way with high content models like organs on chips or this tadpole screen is the future of drug development.
What about using ML for prediction as well? I know you've been using it for evaluation.
No, this is for prediction.
Oh, that was all prediction.
Prediction.
Okay.
No. Okay, example would be, we call it NemoCAD. So you take, let's say the Rett syndrome, okay? You have transcriptomics of patients that have Rett, you have normal, you say across all the genes, 20,000 genes, what genes need to flip to make that normal? And so you basically have a signature, and then you go through databases of every known approved drug or chemical or nutraceutical that has transcriptomic data, and you prioritize them of what would be likely to do that. Then we also create Bayesian network models of gene and gene-drug, with all those drugs, gene-drug networks, based on data that's out there on gene knockouts. And then we ask, which drug would most likely to flip the entire network to normal from disease? And then we do cross-correlations and so on. And we come up with predictions. And then we test them in the in vitro models. And then you could get data from the models and then feed it back and iterate and that sort of thing.
Oh, okay. So that's really
fun. Now, this is all prediction.
Yeah. So I've been playing a lot with using neural networks for regression lately. I don't know if you've ever heard of that, where you can take a neural network that's a big system of weights, and then you can break it down into a kind of expressive equation that describes based on your set of inputs, and then a direct mapping to your outputs rather than just having a kind of mess of numbers in a matrix.
Without the pachinko, yes.
Yeah. And so I was wondering, is there any way to kind of take the neural networks that you're using and find new kind of correlations with these predictions that you've been making?
Well, yeah, I mean, the nice thing about the Bayesian network is you have the networks that you can go back into and get insight into mechanism. And that's led us to come up with new compounds that are even better than the first compounds, or compounds that synergize. Mm-hmm. So that's how we've been doing it, is leveraging insight into the network itself.
So it's a kind of like a nested machine learning approach with an optimizer on top that kind of works with it?
And I'm not the expert on that side. These are my people who are just really talented. But once you build those networks, the gene-drug networks, and now you find out biological activities in these model systems, now you can go in and see, based on the relative effects of all these different drugs, which are the key hubs, which are the key potential targets. We also did things like thermal proteome profiling, where you do proteomics under heating conditions, where you could see, like, with the drug, where you could, and this is where the frogs are actually great, with the drug, and you could actually see which molecule is the drug binding to, and then you could relate that to your network. And then all of a sudden you find the target, and then you find other drugs, and now you could, like, that kind of zooms out, you zoom in on, like, what's important, and now you could think about what other drugs might actually target that in a way that would work, and we've really been really effective doing that. But I want to emphasize, and I've done this for 30 years, work with guys in my lab, bringing people in our computational, you're not going to answer complex biological questions using AI with data and come up with an answer. It has to be this iterative approach with biological testing. And that's where, why not test with, like, human relevant models, or if you're going to do, like we did the high throughput tadpole because we wanted to measure cognition. Now, people always ask me about that. You're going to get a brain on a chip that thinks. No, but I have these tadpoles and they think. And also the thinking is linked to movement. And we're using them to see when you reverse the suspended animation drug, which is called a biostasis drug, are they still functional from a cognitive side? Like, yes, they're alive, but they're autistic now. And so you have to counterpoint different assays to really do this in a meaningful way. The AI gives you predictions, gives you models, gives you networks to probe, but it's not like one equation is going to give you the answer in biology. It's not.
A very large system, very complex.
It's totally environment-dependent biology, okay? Totally microenvironment dependent, macroenvironment dependent, and you will get, the same gene that causes growth leading to cancer is the same gene that causes death, that prevents cancer, depending on the context. So it's not just information. I think that's one of the things that, I've participated in many meetings over the years with computational scientists coming into biology, and they don't get that. And there's a lot of time and money spent just on power of computation with data and information. If you don't have physicality, you don't have biology.
I learned the hard way that a machine learning algorithm is only as good as the quality of the data that you give it. And so if you're missing a lot of these outside forces that in reality you can never replicate something that's going on in a hardware system, you're not going to get a reliable machine learning model.
But other people go the other way, which is you have to know everything in the system to model it, which is totally anti the way physicists model and do pretty damn well. And also in biology, we never know everything that's in it. So the key is you don't have to know everything if you can test and iterate in a biological system, because you might be missing 80% of it, but you might be onto the right 20%. You know what I mean? Or you might find that you can model part of it very well, but not others, and then you know what to look for. So. Right.
Yeah. So you can kind of figure out the little parts that you understand, and
build
those direct inputs mapping to those direct outputs without having an issue there. So another field that I kind of wanted to talk to you about, because you were talking about doing a lot of gene therapies and things like that, is longevity has been kind of a growing field quietly until recently, when you have things like Altos Labs and Life Biosciences that have been working on things like cellular reprogramming, and then other tests of certain compounds that might have to do with epigenome repair and things like this. Have you ever worked with any kind of longevity groups with these organochip systems?
I think George Church at the Wyss has been very interested in this and has been pursuing it. These biostasis drugs that slow metabolism, protect against, looks like they protect against UV radiation, may protect against other sorts of radiation. They slowing metabolism in a sense could be used as a longevity therapeutic. Imagine there was a movie when I was a kid that the guy it was like in like Flint. He wore a watch. He was like a James Bond character. He would meditate, but he would meditate so much that his heart would stop, and then his metabolism would slow, and then the watch would go back and get his blood running. But if each night you slowed your metabolism 10 times what we do now, but you stay functional. I mean, it's interesting. We were talking with groups about longevity recently about that. But we haven't worked on, and we work on so many things that we haven't worked on it in a coherent way.
Okay. I don't know if you've worked with any cellular reprogramming technologies using like Yamanaka factors, but
I mean, we work with IPS.
Right.
But I haven't done that in my own group.
Okay.
I mean, George has a major program where they can identify transcription factors that can be used to make any cell type you want, for example, which is really cool.
So what would you say is kind of like your ultimate goal with these organ-on-chip systems?
I mean, right now we're now using it, as I said, for drug discovery and drug development in a big way. I think biomarkers, like diagnostic biomarkers, we get the flow out of the vascular channel, which is just like taking a blood sampling, and we have done a little with it, but not enough yet. So that's another big goal, is to use that to discover biomarkers that we could then use in our diagnostics as companion diagnostics or predictive diagnostics. We have preliminary work. We have a breath sensor now that is really very specific, so you can maybe think of breathalyzer type things in diseases. So that's really cool. I think the immune system is major, and so now that we have this, lymph node chip, we're beginning to integrate that, linking it to other organs. So maybe you could develop mucosal vaccines, for example. But my group is always, we always keep changing what we're doing. So like these diagnostics work just open up wholly new areas. I just got a Sloan Foundation grant, totally back to my earliest work I ever did when I was an undergrad and grad student, on what's called Tensegrity, which, where I began. I mean, when I was a junior at Yale undergrad, I took a art course, a sculpture course, the same time I took molecular biophysics and biochemistry, where in molecular biophysics, I was learning everything is three-dimensional design of molecules, lock and key, shape. And then I saw these kids walking around campus with structures that look like viruses, which we were learning about in the course, geodesic things. And I asked them, like, what course are you taking? And they said, it's called three-dimensional design. It's like, I got to do this, And my girlfriend turned out, she was an artist, sculptress, was taking a human body sculpture class with the professor who taught three-dimensional design. So she introduced me to him. He was a big Austrian guy, terrifying. He had like lost a leg in World War II. He was really big guy. And she said, this is my friend Don. He's a science major. You'd like to take your sculpture course? He's like, why do you want to take my course? And it was like, like everything in biology is three-dimensional design at the molecular level. What do you mean? And I was saying, like, DNA is like a helix and the shape is important. And he says, what? And I said, like, when you contract your muscles, there's actomyosin filaments that are like two screws that are moving along each other. And he said, you're in my course. And I said, no, really? And I like said, viruses, you're in my course. Just come on Monday. And in that course, and this is a true story, the same week I was learning how to culture cells in a cancer lab, where you would cells would grow on a dish, they flatten, you add an enzyme called trypsin that clips their anchors, and then they'd round up and bounce off the dish. This professor came out and he had this sculpture that was sticks that didn't touch, pulled up against the force of gravity by elastic strings, bungee cords. And as he talked, he would press it, it would flatten, he'd let go, and it bounced off the table. And I was like, oh, that's like... And there was a paper around that time in a science paper that all cells have a cytoskeleton made out of actomyosin that was contractile. So I went back to this is also true, I went back to the cancer lab and we were testing a cancer drug that caused the cells to change shape. And I said, oh, the tensegrity changed. And my postdoc at the time, remember I was undergrad, he said, what did you say? I said, the tensegrity changed. He said, what is tensegrity? And I said, well, it's like I'm learning about it in my sculpture class, like Buckminster Fuller, the architect and the sculptor, Snelson described it. And he said, well, just never say that again. And so I went to the libraries and read up. But that really was a core of the rest of my career. In my PhD at Yale, the last chapter was building tensegrity models of cells. And a Scientific American 1998 cover on this as being the guiding principle of not only how cells are built, but tissues, organs, molecules. And so the reason I even talk about this is, that led me to realize that, well, if, mechanical forces are important at the molecular level for function, and I could see this is how nature builds, it must be important at the cell level. And I had been taking courses in developmental biology from the early developmental biologists. Everything was mechanical. I got into medical school and biochemistry, and there's like, you can't mention the word mechanical. I thought I was absolutely nuts. And so I went to the libraries, I used these models, and that led me to predict how mechanical forces affect biochemistry and gene expression was through this changes in structure, and which I showed in science papers. And so that was really what I'm known for in many areas, is now called mechanobiology. But I haven't really worked on the fundamental science for years, but Sloan Foundation came to me and asked me to apply for a grant that's pure fundamental research and one of the areas was the jump from inorganic matter to the living world. And I had published years ago, based on tensegrity, an origin of life paper where I started with the atom and I went through all steps and got to the first cells, and it was all based on tensegrity plus autocatalytic sets and some basic physical principles. And so we've put in an application with myself, Peng Yin, William Shi, Wesley Wang, and Mike Levin, all at the Wyss. And William and Peng and Wesley have an effort called molecular robotics, which is basically self-assembling molecules. They usually use DNA-type molecules that can be programmed to self-assemble into any shape you want, and they're beginning to do hierarchical structures. My group, the person who does the molecular dynamics simulation, Charles Riley, where we use it to do drug development I was telling you about, that's an interesting story in itself. Charles worked with Peter Jackson, who made the Hobbit films, doing animation years ago, but he also did a PhD in molecular biophysics. And so he took tools from the entertainment industry, animation industry, combined it with molecular dynamics so that we could actually model
molecules in a hierarchical context. We had a paper a few years ago where we basically started with a motor protein called dynein, and then we put it in context of microtubules, which is the sperm tail, and we built up a computational 3D hierarchical model, molecular dynamics, realities, starting with crystal structure, and we could predict how sperms move. So we built a hierarchical model that led to predicting life, starting with molecules. So in any case, in this, we are now proposing. Oh, and then Mike Levin, you probably heard of his stuff because it's all over the press. He has developed xenobots, which are robots of cells that self-reproduce. Mm-hmm. So he's basically rebuilding multicellular organisms that have phenotype unlike anything they had in nature, right? So we're doing top-down, bottom-up of modeling how you go from molecules through hierarchical self-assembly with certain building principles, which Tensegrity came out to be the key principle when we did this from molecule to sperm. And Tensegrity basically says it's a balance of forces where there's a tensile prestress, like isometric tension, when things are pulling together because of attractive forces, but their stiffness comes into balance, and then you bring different structures together and you build more complex systems. But there's always flexibility so that you can change shape a little bit and survive in your environment. It's the way our bodies are built, with bones and muscles and muscle tone determining our shape and stiffness. And so we're not only going to do computational modeling of how these self-assemble, we're building things. So they're building with. William Shih can now build things that start at the molecular level and self-assemble, and you can see them. So they're like millimeter size, but we're going to build scaffolds that then we combine with xenobots that could build artificial tissue. So, that stuff we're doing now too. I mean, it's kind of wild, but it's at the very basis of how life comes into existence. How, what are the underlying guiding principles for self-organization? That's really what the grant's about. Now, where will that go? I mean, well, the molecular robotics group has been doing this stuff. I really supported them because I thought this would be what the Institute would show 30 years out. They had some of the first startups out of it. It turns out these programmable molecules are fantastic for higher specificity, for diagnostics, for CRISPR-based modulation. There are DNA switches that have come out of it. William and Wesley just published a paper where they can use little DNAs as calipers on molecules, so you can do crystal structure in 3 dimensions in solution with hundreds of molecules without freezing them and making a crystal. So in other words, you imagine a molecule in solution, and now you have these little strings that can go from every amino acid to every amino acid, and you can measure their length, which is what they do with these DNAs. And there's enough known about the sequence that you can then come out into the orientation of everything. So you never know where it's going, but we're kind of just following crazy ideas. That's what the Wyss is all about.
So why was Tensegrity such a big paradigm shift at the time? Was the idea before just kind of that all cells were these little blobs?
Yeah. A lot of people think of cells as little blobs, and it's all solution chemistry, when in fact it's almost all solid phase chemistry. That's how you get the efficiency in living reactions. You can't do it in solution. That's how you get specificity. That's how you get location, spatial information, and that's how you interact with the environment. But people were crazed about it. Critics were just, even to this day, the principles of Tensegrity, of mechanical forces being important, pre-stress being critical, that's now accepted, but people still don't use the word Tensegrity. A lot of it's because it was a Buckminster Fuller word, and I won an award at Stanford, and at the award ceremony, the dinner afterwards, like, famous mathematician came up and said, "What you're doing is fantastic, but you can't use that word Tensegrity. That Fuller, he was a charlatan, he was blah, 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 blah." And, you see this in science all the time. Yeah. So I was told at NIH, once I got turned down on a grant, and they said, "If you just don't use that word, well, next time you'll get funded." I said, "Is this like book banning? I can get funding other places. I'm not going to lose my integrity." It's the word that describes what it is.
Tensegrity?
Yeah, it was. A totally true story.
Yeah, I believe it. So, I mean, you are clearly talking about how important interdisciplinary work is at the Wyss. So what are people coming from? I mean, I come from a physics background. Is that something
every background. I mean, my whole career, I would take people
oftentimes because they don't do what I do. In other words, I remember, we were beginning to get into nonlinear dynamical systems, more from a genome modeling type of thing in terms of networks and so forth. And the guy applied as a chemical engineer, got these incredible references, and I'm not sure he never done anything in biology, but he had done like nonlinear dynamical modeling of platinum electrode catalysis or something like that. And he was computational, and I knew we had to get more computational, so I accepted him to my lab. In the beginning, he didn't know what was going on in the room. After 6 months, he was fighting with other people, which meant he learned the language by jumping into a foreign culture. And that's true with everybody. That was what I think why we've always been able to do sort of out of nowhere types of cross-disciplinary things. And he ended up like telling me, he looked at the equations used for photo bleaching and modeling, molecular binding kinetics. He said, this is totally wrong, and then he basically had a series of papers on that, and then, I learned from my students and fellows, and they learn from each other. But at the Institute, you can turn around and find people that do anything from genome engineering to, building devices we have a machine shop with full-time staff where you could build anything in medical grade materials. You can cut through 12 inches of steel. And we could pull anyone in because we're a consortium with MIT, Harvard, BU, Tufts, UMass, all the hospitals in Boston, and other places as well. So it's sort of give us a problem and we'll bring the weirdest set of people to solve it, to bring ideas to it. That's what's really great about it.
So how does an institute like that get built? I mean, dream long-term down the line is that I too one day may be a founder of some kind of style institute.
I gave a lecture, a class day lecture at Dartmouth years ago in medical school, and I said, my life is, people think you move in a line, like especially students applying to grad school, but life is like it's like a biased random walk, right? I mean, a random walk is, molecules moving randomly. In biology, cells do a random walk, but it's biased. So the cell moves back and forth and back and forth. But if there's a stimulus here, like cyclic AMP, it moves back and a little more to the left and back a little bit more to the left, and it ends up going where it wants to be. You just got to follow what you're passionate about and excited about. I really believe that's true. And serendipity comes in again and again and again, and the people you have contact with. But I had done a startup in about 98 when my Scientific American came out on 3D printing of medical devices and other things called molecular geodesics. And we had DARPA funding, and we were going to do tensegrity-based materials that were porous but flexible. The technology wasn't there at all in terms of materials or resolution, but it was really an amazing experience. That's where I learned, somebody at DARPA said, you should not just work on medical, you should also work on non-medical, because if you could bring collaborations with people across Boston, Cambridge that do computational, fabrication, medical, materials, no one can compete with you. And so that was something I learned there. At that point, when the company was doing well, I was thinking about forming an institute on biomimetics, biomimicry. Then that kind of died with September 11th. Then we got involved in this area of biocomplexity, nonlinear dynamical system. And then the HST program at Harvard and MIT was trying to get a plan so they'd get space in a new building, new research building at Harvard, which to this day is called the new research building because they never got funding for it, that maybe the HST could finally have a research space over there. So George Church and I met on a committee to try to envision an institute in bioengineering. And I remember asking the lead, what's the vision, the scientific vision? I said, we're going to have an institute for bioengineering at Boise. So what's the scientific vision? That was it. And so, we came up with a biocomplexity institute idea at that point. Never flew. And then a couple of years later, I was asked by the provost to co-chair a committee with Dave Mooney, who is engineering professor at Harvard, but also was a grad student with me and with Bob Langer years ago when we founded Neomorphics, one of the first tissue engineering companies. So Dave and I co-chaired a committee, and I basically pulled together these two other plans, and I had about a 70% idea of like biomimicry, biocomplexity. George Church brought in. George Church introduced me to William Shee, I think. Dave introduced to other people. I had already been collaborating with George Whitesides and many other people that I brought in. And so we had a group of people, and threw out these ideas, and Dave and I pursued what we called shoe leather, which someone else had suggested to us as the answer to these sorts of building an institute, which is go talk to people. Don't ask them to come to a meeting. Go to their office, throw your ideas, get feedback. And everybody loved this idea, which one day when I was going to a dinner with the former dean of engineering, Venki, I came up with this term biologically inspired engineering, because it wasn't biomedical engineering, it wasn't classic bioengineering, but it basically then got a life of its own when all these other people got excited, and we started to see how they could synergize. And then, we found a benefactor, a donor, we got excited about the vision, and the people that I developed the vision with, we made the first core faculty, and then it kind of took a life of its own. But it really was just, it's like science is like cooking. You kind of fold in things and keep folding them in. And that's people, that's ideas, and that's how it emerged.
So what were some of the kind of pitfalls that you went through? I know earlier on you were talking about how there was a very amorphous idea of an institute, and before you actually
we actually came up with a very coherent idea for the institute very early, because I had this 75% idea. So it was very clear that what the areas would be and who most of the faculty would be. We had some ideas about potential recruits. The challenges with forming an institute are administrative. Well, here's an interesting story. So the donor had come, and we had talked about, a big gift, and then he walked away. He was told by his due diligence input that Harvard's the wrong place to do interdisciplinary research. It's all silos, which is kind of true, all right? And then we kept pursuing the idea. I was lucky to have a house in Martha's Vineyard where he does, and I was able to, reignite his interest and meet with him. But we basically had a vision where, I was able to reconnect with him a couple of years later, meet with him, and he put me in contact with someone who had never been involved in the discussion, but he trusted. And Dave and I met with that guy, and the guy said, I don't know what was talked about before. We threw out the dollar amount. He laughed. But he said, I want to see a risk assessment. And we said, great. And he left, and I looked at Dave, and Dave looked at me and said, what's a risk assessment? And so basically, we put together what included scientific and technical, but it was mostly political, geographical, administrative. And these are things like have and have not faculty. Is this going to create problems with the other faculty? How do we deal with the politics of, are we going to take the best people away from departments? How do we make this a win-win for everybody? Are we going to be on two separate campuses? I mean, literally things like that. Without realizing it, we spent 6 months doing a business plan, designing a structure for the institute administratively that would mitigate those risks. That was the most important thing we ever did. That's the first thing I tell anybody who comes to me is building an institute. And those were then, to this day, the same challenges that are the real challenges of building an institute, which is political.
Yeah, so how did you solve a lot of those problems? How did you figure out how to get certain staff to be on your campus? Or like George Church is still obviously a Harvard professor, but
I am too. I am too. And he has labs. Everybody has split labs for the most part. I mean, that's the plan. So the classic academic vision, and it was for Alston, was that we're going to take these incredible people, we're going to build a building from different disciplines, we're going to put them next to each other, and they're going to start collaborating. Okay, I've seen this. NIH would do this on tissue engineering in the 80s, and they've done it in many different areas. But what happens often is that the people are artificially next to each other, and this, I remember with tissue engineering, the engineers would ask the biologists to teach them how to culture cells and do this, and then they'd thank them in the acknowledgments like they were technicians. The biologists would ask the engineers, can you build me this device? And it was, maybe they'd publish a couple of papers, but it wasn't really collaboration in a meaningful way. So I felt that if you identify problems that are really exciting. You get the best scientists. And they're so hard that those best scientists can't solve them on their own. But you have the really great people there who have the expertise that they would need to collaborate. You just get out of their way. That's kind of what happens with the Wyss. And we don't give faculty lab space at the Wyss. We have collaboratories. So people from David Walt's lab next to George Church, next to Dave Waites, next to me. That totally changes how things work. But, we didn't go to Alston. And if we went to Alston, I was very concerned that you might take a person who's a computational scientist, which we had. She'd go there, she'd collaborate with biologists, do great things, but it would be like an evolutionary branch where she loses contact with her department. So we didn't really want that. So what we did is we basically said, you stay in your own departments, you keep submitting grants, you're on the committees, you do everything you usually do in your department. Your chairmen are happy, but you want to be part of the Wyss, you send over your most entrepreneurial people here. You start submitting grants to here around things together, and you basically, like an MD, PhD, you have to be good at both. It's a struggle. It's more work. You're not, your lab split, but it worked incredibly well. It really worked incredibly well, such that people from both groups can go back and forth, like they can access equipment in either place. And sometimes people start in one, but then they get involved in a project that they think is more translational. They want to do a startup. They come to the Wyss. Other times they stay at the Wyss the whole time. Other times they stay at the home lab the whole time, but they come to lectures and they use equipment. I mean, we're flexible. The key is if we need somebody to solve something, they can come and work on it.
Yeah. So I've heard that you, rather than having grants, you have these things called technology enabling platforms or enabling technology platforms. How does that work? How is that different than?
Well, that's how we started to get up and running. We had 14 core faculty to begin with. We had a 5-year gift with no commitment to continue, $125 million, but it would just die at the end of it if we don't succeed. And the question was, do we have each other take 3 months to write grant applications that we then review each other's application, or have some stranger who doesn't know not as good as any of us to review our application, and then give funding out 9 months later like NIH. I mean, that wasn't going to happen while I was director. So it was like, we created platforms that were led by visionaries. George Church did, like genome engineering, synthetic biology, and Jim Collins, had another platform. Dave Mooney, Joanna Eisberg, I did. And here's a pot of money. You do what you think is the most important thing in your field, but you have to enable other faculty, and it has to be collaborative. And it worked incredibly well. And the faculty who led those were our operating committee, and they were really visionary in another way, which is they were generous. So that when we said, okay, we want to build a new initiative like molecular robotics, but we all have to decrease our budgets by 5 or 10%. We never had a problem. Everybody did. So the platforms have gotten smaller and smaller. Other things have grown. And then we realized that what's most unique about the Wyss is the translational side. And so we've really built out that part, which is much more defining at the Institute, we call validation projects and institute projects. And those have had a huge success rate at going out of startups. It's more focused on young entrepreneurial teams. We bring them together. We have amazing strategic intellectual property attorneys in-house who review reports of invention early and then give you feedback so that from the beginning you're on the shortest path towards impact. You're designing your experiments based on IP and what's going to be transformative. We have business development people of our own who get involved with this process. But the platforms were really covering what we had in the original vision of the grant. I did biomimetic microsystems, which is basically microfluidics and medicine. And that led to organ chips and diagnostics. Dave Mooney started out with nanomaterials that eventually emerged into immunomaterials because that's what they were using nanomaterials for, that had the highest value for the future. Jim Collins started out with, I think it was anticipatory medical devices based in part on work he had done in stochastic resonance and devices that were about, but that's moved into living cellular devices, which is synthetic biology, engineering cellular circuits and so forth. So these have all morphed over 13 years. The platform was to get us started. They're still there underneath, but they're not as central to defining what the Wyss is. So the Wyss now has sort of a pipeline of total open-ended exploratory inventive stuff. Major part of our work is grant funded, but it's usually like either multi-investigator or DARPA, BARDA, IARPA, things that are like breakthrough disruptive technology type things, not R01s. And then these validation institute projects, which are basically de-risking technically, commercially, with the idea of building a team, finding an application, doing what you need to get it out as a startup.
So you said when you had these platforms that part of the deal was that they had to bring in other members of the faculty. How did you make sure that they actually did that?
You know, we never had a problem. It was quite amazing. You know, the productivity from the beginning was awesome, just awesome. And the you know, if you had a graph of intellectual property patent submission, like I had 25 patents, which is a lot before I started the institute. I have 195 now. Okay, so it was like this hockey, and every one of the faculty had things like that. We recruited 40 people from industry with 10, 20, 30 years in product development. We don't have them running a big core facility or lots of microscope. They're integrated with your students and fellows and postdocs and they manage groups, but they also do inventive stuff, and that just turbocharged everything. It helped us understand what the real problems were. It helped us do things, again, on the shortest path towards impact in terms of getting it out from the lab bench into the real world where it's really going to have impact. So there were things that we did that were unusual, like the strategic IP, the people from industry being integrated. Not each platform that you talked about, each one was organized differently because each faculty lead has a different vision, passion, and style. And my scientific advisory board at one point wanted us to use one style. It's like, that's not going to happen because I'll lose my best people, including me, because we all work differently. You can't constrain creative genius. You gotta harness it, right? And so there's a flexibility at the Wyss that is kind of like a kindergarten, and each kindergarten class is different because it has a different teacher. But it works incredibly well. Over time, what we've done is created more structure around these validation projects that are now moving towards translation, and that's because I have a great new chief operating officer who's been in the industry for 20 years, and she's brought a little bit more rigor in terms of it's not so much reviewing these applications, it's supporting and mentoring the people. Like when we have an application for those, and those are the first things you actually have to write applications for. If you get turned down, it's not like, sorry, it's like, okay, how do we help you get something going that could be valued the next time, or could find other funding. And so it's all a very positive mentoring, supportive type process.
Yeah, that sounds like a very different approach. It kind of reminds me, have you ever heard of focused research organizations?
Which one?
Focused research organizations?
No.
Yeah, a student that came out of George's lab, Adam Marblestone. He went on. You might know him.
Wyss. I know Adam. Yeah.
Okay, yeah. So he went on to form Convergent Research. We did an interview with him as well, talking about these focused research organizations, which are basically these nonprofit entities that work with you to try and match funds to have these important ideas that wouldn't necessarily be able.
I've seen mention of it on Twitter actually. Adam was in the early first 2 years at the Wyss, so he probably saw what was going on a little bit. Yeah.
So you've been quoted before saying that the Wyss, it's really important for ideas at the Wyss to get out of the lab and into industry. Why do you think that's so important?
Well, that is our mission, is to have near-term impact. Mm-hmm. As we say, things have to get out of the lab in order to change the world, and academics, I think sometimes that they get a science or nature paper, it changes the world, but that's just the beginning. As I said, organ chips is one example, but the heart, patents are impact, but it's not real impact until people are using it, and that requires overcoming totally different challenges. And I've done 7 startups now, and it's just a different, it's like medicine versus accounting. These are total different challenges, and we try to both nurture people that are passionate about entrepreneurship, but also trying to get them to grasp the realities of the challenges they face. And a lot of these are things like maybe you shouldn't be CEO because you're never going to get the investment that you need. Maybe you should be assistant to the CTO or something like that, so that you get mentored by a CEO who can teach you about like what you have to worry about and what you can do. And but the fact is that the real challenges with a lot of therapeutics or drugs or devices is manufacturing, regulatory, distribution, competition, so you could have the best idea in the world, the best drug, and it never goes anywhere because the wrong company took it. You went to a big company and they had a competing thing, and even though yours, is better in your heart, they were farther along, so they shelved it and it never gets developed, or you bring it to a startup when actually it's not novel enough to be a startup. It should have been licensed to a company who already has a sales force. So if you want to change the world, it has to get outside the lab. And I think that's something that many of the faculty had done entrepreneurial things before, startups and so forth. Some had not at all. And I think that was an incredible experience for those younger people, particularly, who all of a sudden are, licensing things and they're being sold around the world. It really was gratifying to watch that. And it actually is, it's a great challenge to the other faculty to keep up to that level. And everybody keeps, you want to be playing tennis with people better than yourself. That's what the Wyss is like.
That's super encouraging. I kind of feel like, is Emulate hiring? Is the Wyss hiring?
I think both are always hiring at times. Yeah.
So in addition to a lot of these projects that you've talked about, there's even more projects that I read about that the Wyss is working on, you're working on. Some of the ones that I thought were really interesting was nanoparticle therapies that you've been working on, or the special biodegradable plastic. Do you have a favorite project? Do you want to speak to any of those two projects?
We had a science paper a long time ago now, and here's a case where we just have not been able to get the investment to commercialize it, yet we keep getting more and more data. That's exciting. This was, out of my group, the challenge was, what are the biggest killers of man? Heart attack, stroke, pulmonary embolism, all have to do with blood clots and obstruction of a blood vessel. And there are good clot-busting drugs out there like tissue plasminogen activator, TPA. They never became billion-dollar drugs because you only have like a 3-hour window or 4-hour window, and also they're limited by causing systemic bleeding or even bleeding into the brain. So the idea was like, what if you could just target this drug only to the blood clot site? And here's where we went to bio-inspired engineering. It was like, well, how would you do that? Well, nature does that because whenever there's an obstruction of a blood vessel, the shear stress goes up, mechanical again. Right. And platelets are activated by shear stress, and that's why they form a clot in what's called a vulnerable plaque in atherosclerosis, and that's what often causes a heart attack or a stroke. And so we basically made artificial platelets that were artificial not in like a chemist, cell biologist way, like looking like a platelet, but in a physicist way, saying the only thing we cared about is that they would deploy drug where there's high shear stress. And so we used a technique called spray drying that has been used to make drug delivery systems like for aerosol drugs. You take 180 nanometer nanoparticles that are biodegradable. They form into a clump the size of a platelet. And we put tissue plasminogen activator on them, this drug. And we had an animal model where we gave them a clot, inject it. In this case, it was pulmonary embolism. Completely blocked the lung. All the animals die in 45 minutes. We did the same thing, but then we injected these nanoparticles. 85% of the animals survived. But we used 1/100th the dose that you would need to do in a soluble form. It's like a homeopathic almost. That was a science paper. We almost formed a company a few different times, but the BCs, like they were worried about going for heart attack or stroke. The clinical trials, that all the drugs have failed. It's a death ground. Manufacturing of these, you haven't shown you could scale. Long story short, we got funding from the state at one point, and we worked on the manufacturing. TPA is a drug that we didn't have control over. So we then took a drug, nitroglycerin, which everybody's heard about, which has been around for years, off patent, cheap, small, and we just embedded it in these polymers like sustained release, and we showed that we could dilate blood vessels that are constricted really efficiently. And then a guy from Daniel Beard from Oxford University wrote me, and he said,
I work on stroke, but most people treat stroke by trying to open the obstruction or stop inflammation. I work because the one thing that correlates with good response is basically collateral flow around the blockage, like backwards flow. And when you do that, you have much higher flow in a small vessel, and you get very high shear stress. And I was thinking, like, do you think you could deliver a vasodilator? Well, we had just done, we sent him these. It works incredibly well. He's just been submitting a paper on this and presenting a meeting. But we have amazing results in stroke in a rat model. And so now we're talking 2012 was the science paper. So we're talking 10 years. And we're now trying to license it or do a startup again. But that was one of my favorite ones because I thought this is a multi-billion-dollar market. You could deliver, anti-inflammatories for atherosclerosis. You could treat stroke. You could treat heart attack. So anyway, that's an example where the business side of it, we can never go over that hump. The other things you mentioned, we have new stuff that's early but interesting on plastic degradation, where we've identified microbes that degrade multiple types of plastic simultaneously. So it's not just one type. So you can deal with mixed, in a junk pile type of a scenario. There's a startup going out out of Pam Silver's group called Cersei, where they have microbes that can take carbon dioxide as input out of like beer manufacturing and put out foodstuffs, complex fats that can be added to foods. I mentioned the drug discovery type things, but there's so many companies and so many things coming out. I don't have favorites. I'm always amazed at just seeing what's coming out. We had stuff in the robotics area that's already FDA approved. Connor Walsh, wearable robotic flexible suits that basically can, give people who have Parkinson's the ability to walk at normal rate, or it goes the full gamut. Multiple diagnostics companies have come out. George Church, there was IP on CRISPR that was licensed to Editas, that we were early on. Sherlock that has diagnostics for COVID early on. Some of the IP came out of the V's. Emulate came out of the V's. So I think we're up to over 50 companies now.
Wow, that's a high throughput for.
Well, we only have 11 core faculty and 15 associate, all of whom have most of them have their major labs at their home institution. So this is really VIS-enabled stuff. And, Harvard published this, so this was a couple of years ago, and we're basically about the same, if not better, but we were responsible for 23% of all the IP that comes out of all of Harvard University.
Wow, just at the V's.
Yeah, out of the V's, and startups. Wow. Yeah.
So one thing that I've wondered when I've been looking to emulate.
Can I just say, it just shows you that you can do things differently, that the model actually works. It's not just like, every university has lots of startups and this. We only have 11 core faculty. It is amazing. Sorry, you were about to say.
Yeah, but one thing that I was wondering kind of in terms of collaboration, in terms of things with Emulate and the V's. So with a lot of these organ-on-chip systems, I feel like there's a lot of overlap with things like regenerative medicine in terms of iPSCs. And one thing that I've wondered when I first actually started looking into organoids and different organoid groups, I was actually looking into regenerative medicine, not into pharmaceuticals. So I know one of the biggest problems, especially with like 3D scaffolding with organoid systems and regenerative medicine systems, is the amount to be able to culture cells, like the bioreactor problem of not having enough cell volume. Is that something that you guys face with these organoids?
So there are a number of companies. And one thing I have is that all these companies come to us, right? And so there are companies now who have been working on scale up of manufacturing of cells,
right.
CAR T for IPS for other things, and so I think those are things that are being solved in the corporate world, industrial world. That's not something that we've been working on, at least to my knowledge, because I'm about to review our 150 project summaries because we have a scientific advisory board coming up. That's where I learn everything that's going on.
That's a lot of projects.
But there's more than that. But that's not anything we're working on directly I will say, though, that one of the exciting things we have going on at the Wyss is we call 3D organ engineering. Jennifer Lewis and Chris Chen head it. Sangeet Debati here at MIT is part of it. And that's 3D printing, of organs for transplantation.
Right.
And they've been doing amazing stuff with that have a vasculature, a suturable part, and they're already putting them in animals. Looks really, really interesting. And because, I founded with Bob Langer, Xapiens, a tissue engineering company in the 89. And, we put these porous polymers in with some cells. There was one histology picture with 4 liver cells, and they got $2 million to form a company. But the cells die because it takes days to get blood vessels to come in, and you need oxygen and nutrients. So the idea of actually building an organ with the density of cells, as in a whole organ, which Jennifer has published kind of amazingly. It's almost like Frankenstein building. It's not layer by layer. It's like goop building. But also then having vessels that are preformed with endothelium, but also doing it so they get to bigger vessels. It's sort of higher branching. And the last part is suturable. And so you actually could just give it to a surgeon, and they put it in like an organ, things they're used to doing, because that's the guys who are going to be dealing with this eventually.
And they're able to do the suturing with the vasculature. And
yeah.
Oh,
so
wow,
that's what the program started out, and they're getting preliminary, really exciting results. There's just a startup announced recently that's around organoids and with kidney with 3D printing, but that doesn't have the vasculature yet. But this is where the Wyss initiative is going, and they've got some exciting initial results. But there, you're right. The challenge is getting enough cells. Now, iPS are interesting, but you'll notice that like everything in science, there's a peak and a trough of interest. They very rarely do they fully differentiate into really mature cells. They do it sometimes, but most are more like neonatal cells. And so they just haven't gone all the way that I think everybody thought they would. They may do better in vivo, so people are exploring that. I think there's some results that look promising. But that is one way to scale up production. And many companies sell these now, so they must be producing them at huge amounts. The organoids come from patients, and those are grown usually in a gel. And the scale up of that would be helpful. There's one company I know that produces some, but that's an area that needs development. We haven't been working on that, but that's an area that needs development. And I just had a conversation with Chris and Jennifer yesterday just about the need to work on that for their particular application, because you can't have this beautiful thing and then not be able to build enough to do a clinical study.
So I know the Wyss, you're mentioning a lot of the projects at the Wyss. Does Emulate do any regenerative medicine work, or is it solely just organ on chip?
Any company has to be really focused right now. And so they work with corporate partners what their needs are. So they may be working on things that I'm not aware of, but I don't think so. And then it's mostly developing products they can sell and developing sales forces and training forces to help people make sure they get successful results. But they've done a lot collaboratively with groups.
So as probably one of the last questions is more of a philosophical question I'm looking at. So you've talked a lot about using these organ-on-chip systems to do things for preventative medicine, but also for preventative work on viruses and kind of pandemic prevention. How concerned are you about existential threats? We've had talks with people like Kevin Esfeld, who we hope to have
Kevin
on the
was
show.
at the Wyss, as you know.
We've talked to him informally. We haven't had him on the show yet. We've been trying to schedule him.
Was a great. Gene Drives was developed at the Wyss. It's our IP. It was really.
Oh, I didn't realize it was at the Wyss. I knew it was through Harvard.
I never realized that. It was all our IP, the initial stuff.
Oh, wow. Yeah. So, but getting back to it, how concerned are you about certain existential threats like pandemics turning again? Are you working to prevent these things?
We have major efforts. I had a $16 million DARPA grant around COVID. I now have another grant from DARPA around COVID. I was funded by NIH around respiratory pandemic viruses and DARPA before COVID ever happened. That's why we worked on influenza. And so I view this as a continuing challenge. It's going to happen again and again and again. So, we're working on an oral drug that works against all coronaviruses. and another one that works against all influenza, regardless of type. And we're making major headway. We just got a big gift from Open Philanthropy on those. And so far, the one against COVID works against, at least in a pseudotype model, it targets the spike protein against every variant that we can get our hands on, including Omicron, as well as MERS and SARS-CoV-1, common cold virus, coronavirus. And this is an oral drug that it's a modified version of an existing drug that's novel, but it has similar pharmacokinetics, meaning it's oral and you get very high concentration in your blood. So we're moving to animal testing, but like that could be something that is a preventative drug. And that's exciting to me. It could be cheap, it's oral, small molecule. So that's an example of what we're working there. I think we're working on, Jim Collins has done amazing things on wearable masks. We have our multiplexed electrochemical sensors. I mentioned earlier, you could mention viral load, RNA, protein, host antibodies, again, could be used in future pandemics. I'm funded by the FDA around radiation countermeasure drugs. It's hard to test drugs against lethal radiation in patients.
Right.
Animals don't really replicate humans. So we're doing human and non-human primate models on organs on chips right now, and we're looking at drug development there. So that would be, other, call it existential threats. It's getting more real given what's going on in Ukraine today. Then there's there's biological threats, obviously, that could be engineered things. I think a lot of the diagnostics that we develop could be useful for that. The diagnostics we develop in my group are portable. So these electrochemical glycoglucometer, this could be done at home or in doctors' offices, in pharmacies. But was there something else that when you said existential you were thinking about? Things that you can't even imagine now? Is that what you mean?
Well, I was thinking more pandemics was the obvious one, but also, like you mentioned, nuclear warfare. And then the third one that gets brought up again is machine learning and AI.
Oh, you mean AI in terms of taking over?
Right. That's when I heard that in college originally, I kind of laughed at it.
As the director of the Institute, I am legally responsible. So I'm always worried about, I was worried about gene drives when that came out, when Kevin was there. I'm worried about when we do viral evolution of viral mutants. That's terrifying. But we have a really good safety officer full-time on site. We really try. We have an ethicist on site, works with us to think about kind of ethical issues. Things like, now the xenobots and reproducing robots that are, cellular. If we do this stuff between non-living, living world, what does that mean? In terms of can you generate, living entities or self-replicating entities? We're not there yet, but we try to do it in a thoughtful way. And as soon as something happens like gene drives, and we did a very thoughtful, written pieces. George and this ethicist and the whole team, Kevin, before it came out about what the concerns are, how it should be handled. Unfortunately, not everyone, once these things are out, people, now with synthetic biology, people can do this anywhere. But we've tried to handle it in a really thoughtful way and a proactive way.
So in kind of you've given us an overview, starting a lot about talking about what you found when you were in college and then about in grad school and things like that. What about when you were even younger? Did you have any kind of formative experiences or interests that kind of pushed you towards this path?
Well, that's interesting. I mean, I always, my friends' parents would always get me science toys for my birthday. Yeah. And I didn't want them. I wanted building sets, Legos and Tinker Toys and Erector sets. And so they must have seen something in me, right? I always was very good at explaining how things work intuitively. So I remember my little friends asking me how could that work? And I just would give an answer, and then they'd look at me and say, oh, that explains it. And I also was very mechanical. I would take apart television sets and put it back together again. And when I was in high school, my guidance counselor, they gave you tests like achievement, IQ tests, achievement, whatever, to tell you what you'd be good at in your career. And they said, you'd be really good at automobile repair and, air conditioning maintenance because you scored off the charts in mechanical reasoning, like 3D. So, you ask, I definitely like science. I read about scientists and things like that, but I like patterns in nature. I just like to learn. I like the creative process. So I enjoyed everything at school. I always did well at school. Some of the most impactful things in my life, you ask me, I got into a program where I did 4th, 5th, and 6th grade in 2 years. I had special teacher. There were only 12 kids, special art teacher, special music teacher. We do things like read, Treasure Island. We'd watch every movie ever made of it. We would build puppets. We would build a stage. We'd write a play. We'd put it on for the rest of the school. And we do that kind of interdisciplinary thing for everything. It's one of the most stimulating experiences of my life till I got to Yale. Yale was transformative because all of a sudden I went to public high school. My parents never went to college. I grew up in Long Island in New York. I call it Mesoamerica, the middle class and the town had 60,000 people, but it was an unincorporated village. It was just houses, identical houses pretty much. Yale was transformative, but when I got there, I got in on math. I was really good. I was a math lead. I went all the 800s on everything, advanced placement, and but it's another true story. I sat in on a class and I forgot what it was called, but it was way up in math because I had all this advanced. And everyone there had big, thick glasses and was incredibly serious. And it was like, I said, I walked out of there and I never took math again. And then I also was so excited about history and art and music and courses that you could take that I didn't really take science the first semester. But then I got into classes that got me turned on, and basically I ended up doing combined bachelor's and master's in molecular biophysics and biochemistry. Now, why molecular biophysics, biochemistry? If I was interested in all these different areas, well, it turned out that I did a BA, not a BS, and an MA, and that allowed me to take a lot of courses in any department I wanted because I already had advanced placement, and I could take almost graduate level courses for a lot of the MB&B. And then, so that let me take an incredibly varied type of education, and at Yale I did theater, I did all sorts of things, and all my friends did different things. Like when we did a play, there was nobody who was a theater major. There wasn't really a theater major. They were scientists, they were philosophers, they were sociologists. So, and then I just basically stayed in academia the rest of my life. So, and I stayed at Yale for 11 years, and I knew every library. And basically, that liberal arts type of thinking where people just did what they were passionate about but didn't worry about the discipline. I've been lucky to stay in that, going from Yale to Harvard, and I feel really lucky about that. And that's so to me, I kept the interdisciplinary thinking of undergrad when I was a grad student and went on from there. I will say that I see that in people who've come to work with me, that if they start working and come to me as an undergrad, they're totally flexible in terms of interdisciplinary. They come as a grad student, they've already narrowed down a little bit. If they come to me as a postdoc, they've narrowed down more. If you come as a visiting professor, they've narrowed down more. And so it's keeping that kindergarten-like mentality. And the scientists I see that aren't like at the Wyss, they really like kindergarten kids. And they'll work on anything. It's not like they work in a field, like this is my boundary. And very few of us were trained in engineering, yet we're leading bioengineers now, right? George Whiteside is a chemist, but he developed, microsystems and microfluidics. George Church was a geneticist, but he developed all this automation that does everything now in terms of genome sequencing, genome synthesis, and it's a largely equipment, AI, machine learning. He's trained in genetics. And Jim Collins was systems theory in control theory in engineering, and he's a leading synthetic biologist. You follow your passion. That's the answer.
So kind of leading out after that question would be, so obviously everything at the Wyss that you described is super cool and super interesting. But why do you think it is that a lot of the work that you work on is super important? Like, why would you, I mean, you talk about drugs and things like that, but say I was some wealthy philanthropist, why would I want to give it to something like the Wyss rather than, I don't know, some other program that's...
I think it's really simple in that we take a different approach. We're problem focused. And so you give me any problem and we will bring the most, it's like a SWAT team, like a dirty dozen, but of the best in terms of being creative, and bring this incredible, palette of colors that you can paint things that no one's ever seen before. So we kind of do out of the box sort of stuff. If you go and give money to the experts in an area, they will do incremental advances based on what all the other experts and they have developed in the past. If you give to people who are just incredibly creative and have incredible capabilities, and you give them a new challenge that they get excited about, they will do something new. And I had a big food company come to me, and they were saying the areas that we, in 5 minutes I came up with an idea of doing things that brought synthetic biology, drug delivery, and all different things together in the food industry. And when we got a big funding right away.
That? I'm curious, in the food industry.
I don't know how much I can disclose.
Okay.
But it deals with, it's a creative way of dealing with getting sugar out of, bad sugars like fructose out of food without changing the manufacturing of them. That's all I will say. But anyway, we're talking to people now about Alzheimer's disease. You look at the Wyss, it's like we don't focus on Alzheimer's disease. I looked around at what we do. We have like pages of what we're doing on things relative to Alzheimer's disease. Diagnostics, we're blood-brain barrier models. We're developing shuttles that bring drugs across the blood-brain barrier, which is like a major limit for all brain diseases, because only 1% of the drugs get across. That's one of the big problems. Just things that are under there where creative people in the lab are just playing. It's like, oh, I'll put this would be useful for that. It's just like, oh, I'm cooking and I can mix this with that, and that might be good as a dessert too. That's what science really is. So I mean, I really believe that philanthropists are either going to give money because they want to give money to a famous name and they know that it's going to be safe because they'll continue to do what they've done well, or they can do it to be disruptive and really change the world. Hans-Georg Wyss is somebody who wanted to disrupt not only the science, but how Harvard University worked, how academia worked, crossing the academic industrial boundary, and that's what we did. But we've done that with DARPA funds. They're the venture arm of the government, and they're trying to solve impossible problems, not difficult, but impossible problems, and think 30 years ahead. We have a huge amount of DARPA funding at the institute. I've been funded consistently since 1998, and I have sometimes 3-4 DARPA grants at once, But they're focused on solving a problem, not just showing you published papers. And that's what we do at the Wyss. I think that's philanthropists who really want to solve a problem and to do it in their lifetime. That's why they should come to us, I would say.
Well, it's been great talking to you, Dr. Ingber. I was so excited to have you on. I think this has been a great talk. Thank you so much for coming.
Pleasure. Thanks so much.
Yeah.
Okay.